Howdy, my name is Luis Hurtado, and today I'll be telling you about the origin of the western honeybee, Apis mellifera, and specifically why evidence from mitochondrial genomes, or mitogenomes, suggests that this species originated in northern Europe. For context, Apis mellifera is a species of cavity-nesting honeybee that is originally native to Africa, Asia, and Europe. It has since been introduced throughout much of the world, including North America, South America, and Australia, because humans were able to domesticate these bees and rear them to produce honey and other products, and because they are super effective pollinators that help support a third of the crops we consume. Recent taxonomic revisions have suggested that there are 33 subspecies of Apis mellifera, although the exact number is debated. For several decades, scientists have been interested in elucidating the phylogeography of these subspecies. Phylogeography is the study of how evolutionary related lineages are distributed across space and how these distributions came to be over time. One of the things we can predict using phylogeography is where the common ancestor of a group of lineages lived, which represents the geographic origin of the group. Evolutionary relationships in phylogeographic studies are generally inferred from nucleic acid sequence data, and mitochondrial DNA sequence data is often leveraged towards this end. If you remember from high school biology classes, the mitochondria is an organelle used primarily for energy production. It also has its own DNA that is inherited independently from DNA in the nucleus. In most animals, mitochondrial DNA evolves more rapidly than nuclear DNA, so it can be really useful when comparing closely related groups, like subspecies. Knowing this, the big question I had was, where do mitochondrial genomes suggest Apis mellifera originated from? To start off, we must first verify that the Apis mellifera subspecies are more closely related to each other than to any other bees within the genus Apis. We can see that in this phylogenetic tree, which was inferred from mitochondrial genomes, Apis mellifera subspecies form a monophyletic clade, meaning that they are all descended from a common ancestor and that no other taxa that are not recognized as Apis mellifera subspecies are included in this group. Next, we see a phylogenetic tree inferred from mitochondrial DNA that shows Apis mellifera subspecies only, so that we can more closely explore the relationships among these. These lineages are color-coded based on their geographic affinities. Black represents those found in Northern Europe, green indicates those found in Eurasia, blue shows those found in the Middle East and Northern Africa, pink and purple show those found in Eastern Africa, yellow denotes the Mediterranean region, and red represents sub-Saharan Africa. The important takeaway from this figure is that Northern European subspecies became reproductively isolated from the rest of the lineages early on in the evolution of this group, and the next major divergence happened between subspecies found in Eurasia and those that are presently found across Africa, the Middle East, and the Mediterranean. Given the information from this phylogenetic analysis, we predict the following dispersal pattern. On the left is the phylogeny I showed previously, and on the right is the predicted dispersal pattern. Because the Northern European bees are most distantly related to all other subspecies, we infer that the common ancestor of all Apis mellifera was found in Northern Europe, and that the first process to take place was that some bees split off and started moving southward. From this point, further splits happened between bees that would colonize much of Eurasia and those that would colonize Africa and the Middle East. This is further supported by the fact that many of the African subspecies appear to have split from each other within recent evolutionary history, so it would make sense if they arrived onto that continent relatively recently. Another thing I want to highlight is the placement of subspecies Apis mellifera iberiensis, which you can see in the yellow Mediterranean group. This lineage is technically found in Europe, but based on the present study, it is more closely related to bees found in Africa than to Eurasian or Northern European bees. This suggests that it originated from African bees that returned to Europe, and I bring this up to show why it is important to be specific when elucidating the origin of lineages. Hence why I specify that mitogenomes suggest a northern European origin for Apis mellifera. Based on the data I've shown you, there are several important takeaways. The first is that the Apis mellifera subspecies appear to form a monophyletic group. The second is that northern European bees appear to have become reproductively isolated from the rest of Apis mellifera subspecies quite early in the evolution of this group, suggesting that northern Europe represents the location that the most recent common ancestor of all the subspecies inhabited. Subsequent divergence events happened as some bees moved southward into Eurasia and Africa, where a lot of recent diversification has occurred, and we have even seen one subspecies make a return to Europe through the Iberian Peninsula. All things considered, this information provides strong support for the Northern European origin of Apis mellifera based on mitochondrial genomes. Over the past several decades, multiple competing hypotheses have been proposed concerning the origin of Apis mellifera, with mixed support for each depending on the method used. The present study incorporated more information than previous works and found strong support for a Northern European origin. But it is important to recognize that at the end of the day, these are inferences and that there is no way of knowing what happened with 100% certainty. Nonetheless, this work greatly contributes to our understanding of Apis mellifera phylogeography, a topic of great interest since the mid-20th century. From an applied perspective, findings from phylogeography can help us make decisions about how to use different bee subspecies in apiculture, as it provides hints about why these lineages may have evolved the adaptations they exhibit today. Additionally, this knowledge may help us make informed decisions if we are interested in crossing different subspecies to create hybrids that express combinations of favorable traits for specific contexts. With that, thank you for watching.